Okay, so this is a video review for your test 2. As a disclaimer, I want you to note that anything that was mentioned in class or lab may be tested unless I've specifically said something in Acorn or in class. The test is approximately 50% multiple choice and 50% short answer. The lab material isn't reviewed here, but is testable material, so make sure that you review your notes. Where do T cells develop? What are the stages of T cell development? So T cells develop in the thymus. They originate in the bone marrow, but they do develop in the thymus. And the stages or main big picture stages of T cell development is that they go through a period of time where they're double negative, meaning that they don't have CD4 or CD8 on their surface. Then they rearrange their TCR genes, and then they become double positive for both CD4 and CD8, and then depending on whether or not they interact with MHC1 or MHC2, they will become single positive for CD8 or CD4 cells. What pushes hematopoietic stem cells to become T cells? So one of the early ligands that help push these thymocytes to become a T cell is Notch ligand binding to the Notch receptor. So bone marrow stem cells, as they come to the thymus, these are precursor cells. They're still a stem cell, and so if they're given different growth factors, they may turn into a B cell or a T cell. So Notch is important. Notch ligand binding to the Notch receptor on that stem cell is important to help generate a T cell. When does the T cell receptor genes get rearranged in development? So this happens, it begins in the DN2 stage, and it's finalized during this double negative stage. So it's in the double negative stages of development that the genes are rearranged. Which T cell receptor are the most common? So we most often see the TCR alpha beta. The beta rearrangements are the ones that occur first, and they tend to be, so that's the receptor that's going to be most productive. So we do see TCR alpha beta being the T cell receptor that's chosen most often, more so than the gamma delta. Once TCR beta is rearranged, what happens to it? So TCR beta rearrangement occurs first, and when it produces a successful chain, it will go with a pre-T alpha chain, so a protein surrogate to the surface of this T cell, giving you a pre-T cell receptor. When is the T cell double positive? The T cell is double positive not until it has a T cell receptor on its surface. So not the pre-T cell receptor, it's not until it has a TCR on its surface. What is different between positive and negative selection? So when we look at T cells and how they go through positive selection, this is so that they'll interact with and bind to MHC molecules with low affinity. And if they bind to self-MHC with peptides with a high affinity, that's when they're negatively selected for, and then will die by apoptosis, because we don't want to have T cells that are binding strongly to self-antigen. We would want to have those killed because they are responding to self-antigens too strong. We do want to have recognition of the MHC though, and so we need to have some positive selection so that we can get our T cell receptors recognizing our own self-MHC molecules. How do T cells become single positive? So it's thought that they go through this process where they end up becoming single positive for CD4 or CD8, depending on whether or not the T cell receptor that it has binds to a given MHC molecule. So if the T cell receptor binds to MHC class 2, it will keep its CD4 molecule, and if it binds to MHC class 1, it'll keep the CD8. And so as you look at the T cells and the T cell receptors that they have, if they bind with a high affinity, they're going to be deleted because we don't want self-reactive T cells. Some of the highly reactive will become Tregs, so they'll be important for having tolerance in the periphery. Some of them that bind with an intermediate affinity will survive, those will be the ones that get positively selected for and kept. And the ones that don't bind well at all will die by neglect. And that is the majority of the T cells do end up dying. Over 90% of them do. How are T cells able to not respond to many self-antigens? So they in the thymus, there is this protein that's made, the autoimmune regulator, this AIR protein, and it expresses proteins that are normally expressed in different tissues so that you can get a lot of different types of proteins presented to these T cells in the thymus. And so because of this is how we can get them to be less self-reactive in the long run. How are T cells able to leave a lymph node? So they are able to leave after they've increased their sphingosine 1 phosphate receptor, which is required. This receptor will bind to sphingosine 1 phosphate and will help it to actually end up leaving the thymus. This is true for the thymus. It's also true for how they get out of the lymph nodes as well, which we've looked at. What mechanisms promote tolerance to self? So you can decrease cytokines, so depriving cells of cytokines can promote tolerance. There are cytokines that are inhibitory, such as TGF-beta, which can inhibit T cells from being activated. You can also have the inhibition of antigen-presenting cells and the killing of T cells, so cytotoxicity to the T cells. So there are multiple mechanisms to try to have your immune cells to not respond to self-antigens and therefore be self-tolerant. Where do B cells mature? They they develop in the bone marrow, they originate in the bone marrow, and they do go through their gene rearrangements of their immunoglobulin genes in the bone marrow, so that all happens in the bone marrow. As they're leaving the bone marrow, they have IgM on their surface, but they're an immature B cell. So B cells actually mature in the spleen, and that's where they become mature. And you can tell they're mature because they have IgM and IgD on their surface. What is an important growth factor for B cells? So an important growth factor for B cells is IL-7. So that's a cytokine that's made, and it binds to receptors, the IL-7 receptor, which signals through JAK-STAT signaling to end up giving this B cell pro-B cell proliferation signals, also promoting the gene rearrangements and overall survival signals for this B cell. So that's important for the B cell, an important growth factor for B cells. What is the B cell called if the heavy and light chains have rearranged and it has IgM on the cell surface? So that would be called an immature B cell. It has IgM on its surface, and it has heavy and light chains. If it doesn't have a B cell receptor on its surface yet, it has a pre-B cell receptor, we would call it a pre-B cell. They are pro-B cells when when they're starting to rearrange their heavy chain genes. How can you tell if the B cell is mature? So it's mature when it has IgM and IgD on the surface of it. What are the subsets of B cells? So we've talked mainly so far, and we talk most often about follicular B cells, which are called the B2 cells. They are B2 subset. There are other B cells that exist. So the B2 cells, the ones we talk about most, and if I don't specify what type it is, I would assume that it is a follicular B cell. These are the ones that exist in the secondary lymphoid organs, and the B1 cells and marginal zone B cells occur in different locations. So the B1 are in the peritoneal cavities or pleural cavities, and the marginal zone are in the spleen. So there are some differences on how they get activated and how we can detect them. So, for example, the molecules that are on the surface can be different. Both have IgM and IgD, but the B1 cells have CD5 on their surface, and that's one way we can detect them. There is somatic hypermutation that occurs extensively with the follicular B cells, but very little with the B1 and the marginal zone B cells. And the follicular B cells need T cell help, whereas the others need very little or no T cell help. How are T cells activated? So T cells are activated when they are presented antigen to their T cell receptor. So if it's a naive CD4 positive T cell and it's presented antigen in MHC class II to its T cell receptor, it will be activated through that signal along with co-stimulation. This will cause that T cell, if it recognizes that particular antigen, to make cytokines, and that cytokine will act on the T cell itself as a growth signal. IL-2 is important for growth of this T cell and ultimately clonal expansion, which means there'll be more of this T cell that recognizes this particular antigen. These cytokines are also important for CD8 positive T cells. CD8 positive T cells will bind antigen presented in MHC class I to its T cell receptor, and with co-stimulation it will be activated along with the help of cytokines that come from the activated T helper cells. They can make some IL-2, but the cytokines from the CD4 positive cell that's activated will help further promote this CD8 positive T cell. These cells, once some of them will become effector cells, so the CD8 positive ones, the cytotoxic T cells, will go out and kill infected host cells, and they'll have memory cells so they can respond quickly a second time you come in contact with the same pathogen. They'll also have, the CD4 positive cells that expand, their functions will be to help other cells. They can help macrophages, other B cells, and the memory cells will circulate and to be around to help if they need to respond quickly another time you come in contact with this. What's required for T cell activation? So we need two signals. There's a two-signal hypothesis. Signal 1 comes from the T cell receptor signaling, and the second signal comes from co-stimulation. So CD28 on the T cell receptor and B7 molecules such as CD80 and CD86 on the antigen presenting cell. The third signal can come from cytokines that will help direct the type of T cell that you're going to get here. What's in the immunological synapse? So inside the synapse, we have at the center the T cell receptor, which is binding to antigen and presented in MHC. So if it's the CD4 positive T cell, it's with MHC class II. CD4 is important for helping to recognize that MHC class II. Some other things that are important here is that CD3 is involved. So that's similar with the T cell receptor on a CD8 positive T cell. There's CD3. It's not CD4 though, it's CD8 that's recognizing the MHC, and here it's MHC class 1. So there's some similarities and some differences between the center of these synapses. If you were to look at the outer parts of the synapse, there are some similarities. Co-stimulation comes from CD28 binding to CD80 or 86 on either the CD8 positive T cell, that interaction with the antigen presenting cell, or on the CD4 positive T cell. And then you have cell adhesion molecules that strengthen this on the outer parts. So CD2 on the T cell with LFA3, and LFA1 with ICAM1. So those are cell adhesion molecules that basically help make this a much more stable interaction. What's going on during T cell signaling? So when we looked at T cell signaling, it starts here up with this T cell receptor, and CD3 is important for it to occur. And because it has these ITAMs on it where they can get phosphorylated, one thing that's important here is that you get the phosphorylation of these ITAMs on the CD3. And so there are kinases involved. And the first kinase that is LCK. It's a tyrosine kinase, which will phosphorylate these sections of the CD3 when the T cell receptor is engaged. And so this induces a cascade of events leading to multiple different molecules being phosphorylated. In the end, you get transcription factors going into the nucleus and changing the expression of genes that are important for that cell to survive so it can proliferate and then have different effector functions. What are the co-stimulatory molecules? So co-stimulation, positive co-stimulation comes from CD28 or ICOS. And negative co-stimulation can come from, we've looked most closely at CTLA4 and programmed death 1. And those turn off these receptors and inhibit them, whereas the positive co-stimulatory molecules help activate these cells. What happens if there is no co-stimulation? So if there's no co-stimulation, the cell is non-responsive. So in an activated T cell, it gets signal 1 through binding antigen in an MHC, and signal 2 is the co-stimulatory molecule. If this T cell over here doesn't get that co-stimulation, it's going to be non-responsive or anergic. It can also be anergic if it gets those inhibitory signals being promoted, so that they'll inhibit the response and overall be a T cell that's non-responsive. What are superantigens? So superantigens can basically short circuit the need for co-stimulation, and even if an antigen is being presented by a given antigen presenting cell that the T cell receptor doesn't actually bind to, a superantigen can still activate this T helper cell to proliferate and produce a lot of T cells that are inappropriate. So this can be helpful for the pathogen so that you have a T cell response going in the wrong direction ultimately. The superantigen binds to the T cell receptor, the beta chain and the alpha chain of the MHC molecule. What happens when T cells are activated? So when a T cell gets its two signals, signal 1 from T cell receptor binding its antigen in MHC, signal 2 from co-stimulation from that antigen presenting cell, it's going to proliferate. So it's going to clonally expand. IL-2 is a growth signal that helps push this through the cell cycle, and you'll get many more cells of this with that exact same T cell receptor generated. So you have a clonal expansion of these cells. What does signal 3 do? Signal 3 comes from those cytokines that can be polarizing. So an antigen-presenting cell that's been activated by whatever it's coming in contact with, it'll secrete things like cytokines that can help tell the T cell ultimately what kind of infection is going on. And those cytokines will polarize the T cells to make their own certain set of cytokines. And it will help turn on certain genes so that that cell will stay making these certain cytokines. What type of different T cells are promoted from these polarizing cytokines? So we looked at how different cytokines that are made from antigen-presenting cells can promote a naive CD4 positive T cell become one of many different effector T helper cells. So if the response that's being given is anti-inflammatory and making things like TGF-beta and IL-2, you'll get the generation of Treg cells, and that inhibits immune responses. If you have cytokines that are made that are pro-inflammatory and trying to get rid of an extracellular pathogen, you will have IL-6, IL-23, and TGF-beta promoting a Th17 cell, which makes IL-17 and IL-22. And the transcription factor in these cells that's predominant is ROR gamma T. In the Treg cell, these natural Tregs, or these induced peripheral Tregs, a common transcription factor in them is these FOXP3. So we can identify Treg as having most often FOXP3 as being expressed quite high. If the conditions given towards the T cell include IL-4, you'll get a Th2 response, which the transcription factor is GATA3. These Th2 cells produce IL-4, IL-5, and IL-13, and they're important in worm infections, and they do have a role in allergy as well. Another cell that has an important effect in worms and extracellular pathogens are the Th9. And so with IL-4 and TGF-beta, you can get these Th9 cells generated, and they make a lot of IL-9. The follicular T helper cells, you have the transcription factor BCL6. They're promoted by IL-6 and IL-21, and they can make IL-4 and IL-21, and they will help the generation of B cells and the and maturation of the germinal centers. So these T cells are important for interacting with B cells in the germinal center. T helper 1 cells are important for intracellular pathogens, and so the cytokines that would be made by that antigen presenting cell to polarize it to be a Th1 are IL-12, interferon gamma, IL-18 together, and these T helper cells make characteristic cytokines such as interferon gamma and TNF. This is really important for activating macrophages to work better and to activate the other cells to be able to get rid of an intracellular pathogen. Another T helper cell that helps for extracellular pathogens and importantly in the skin are the Th22, and they are generated from those the cytokines IL-6 and TNF, and they actually make IL-22, and this seems to be important in the skin. We're going to focus most on the T helper 1 and the T helper 2 cells, so make sure you know the transcription factors and the cytokines involved here, and overall the big picture of the types of pathogens that they're going to respond to. We also look at throughout a lot of the follicular T cells because they come up a lot with respect to helping those B cells. Another one that we will talk about because of extracellular pathogens is Th17, and we did talk about that a little bit in the lab as well. and peripheral Tregs, and the fact that FOXP3 is a common transcription factor there will be relevant. What are key differences between Th1 and Th2 responses? So the type 1 response, giving you a T helper cell, it will come when you have an antigen presenting cell giving signal 1 and 2, and then the signal 3 comes from these Th1 polarizing factors such as IL-12. And so when that cytokine binds its receptor on this T helper cell, it will activate the production of the transcription factor TBET. And this being turned on helps this become and be a Th1 cell, which will then make a lot of interferon gamma. If the conditions are more of a type 2 response, meaning you need a humoral immunity, so this is also important for pushing towards antibody responses and extracellular pathogens such as worms, you have an antigen presenting cell giving signal 1 and 2 to this T cell, and the types of cytokines are Th2 polarizing factors such as IL-4, which give this T cell a signal to make the transcription factor GATA3. And when GATA3 is turned on in this T cell, it's going to be a Th2 cell, meaning that it's going to make cytokines such as IL-4, 5, and 13, which can help for responses to worms as well as humoral immunity. How do T helper cells actually help? So they help often by making cytokines. So the example here on the left is a B cell helping a B cell. A B cell will get activated when it binds to antigen on its B cell receptor, and then with T cell through CD40 ligand binding to CD40, it'll get another signal, as well as the cytokines that are made here will help to influence this B cell to proliferate, so there'll be more B cells, also help it to switch which isotype it's making and go through affinity maturation, ultimately helping determine which type of antibody is made here. T helper cells can also help CD8 positive T cells, and so a T helper cell, when it gets presented antigen in MHC class 2 and gets co-stimulation, it will start making cytokines. These cytokines can have effects on a CD8 positive T cell that's also seeing antigen in its TCR through presentation through MHC class 1. And so this T helper cell is helping the CD8 positive T cell to proliferate as well, so that it will make functional and proper memory responses. What is the clonal selection hypothesis? So this means that you would only clonally expand the one particular cell that's responsible for recognizing the pathogen that you currently have. So in the bone marrow, you have stem cells. There's gene rearrangements that occur to give you many different B cells. We're only drawing 4 here. If they don't respond to anything, they'll be deleted. If they do, or if they don't mature in the spleen, they'll be deleted. And if they do mature, so they have IgM and IgD on their surface, and they respond to antigen, only that clone will multiply so that you can have a proper response to the actual antigen that you are being presented with. What is different about antigens that are T-dependent versus T-independent? So T-dependent antigens tend to be proteins, and T-independent tend to be long things like multivalent or polymerized antigens, and it tends to be more often proteins that are T-dependent. What is different about T-dependent and independent antigen B cell activation? So for T-dependent, basically when it's a protein, it'll get signal 1 for B cell receptor binding antigen, signal 2 from the T helper cell binding CD40 from CD40 ligand, and it'll get signal 3 from cytokines that are made from that T helper cell. T-independent antigens can be activated through antigen binding their B cell receptors, or a second signal coming from a pattern recognition receptor on the surface of that cell. It can also get signaling with the help of the complement receptors along with the B cell receptor to be activated if it's a T-independent antigen. What happens when you have T-dependent antigen activation of B cells? So T-dependent activation of B cells, you get signal 1 from the antigen binding the B cell receptor, signal 2 from the T helper cell, signal 3 from the cytokines. You'll get some of these cells proliferating and becoming memory cells. Some of them are going to become plasma cells and start making antibodies. And some of them are going to go into the germinal center and start trying to make antibodies that will bind with a higher affinity. So they'll go through somatic hypermutations and class switch in here. What co-receptors propagate B cell receptor signaling? So it's for the B cell, they can have CD19, CD21, and CD81 help propagate the signaling through the B cell receptor. If we look at the B cell receptor and its signaling pathway, there's also phosphorylation events, so kinases are important at the beginning of the signaling. The intracellular tails of the B cell receptor itself isn't long enough, so there are other molecules, Ig alpha and beta, that are phosphorylated here, and CD19 that are important in initiating these cascades. These pathways ultimately result in the movement of transcription factors from the cytoplasm into the nucleus, and they then change the expression of the genes that are in the nucleus, and they turn on genes to help promote this B cell to survive, to proliferate, and ultimately differentiate into the B cell that it wants to become. When a B cell is activated, what chemokine receptor increases and what result happens? So when a B cell is activated, it increases its expression of CCR7, and this helps it, so the result is that it helps it move to this boundary of the B cell to T cell zone, so that this allows it to then interact with T cells, which it needs T cell help. What transcription factors help determine the fate of B cells? So if a B cell is making IRF4 and BLIMP1, it's going to become an antibody producing cell, so a plasma cell. If a B cell has high levels of PAX5 and BCL6, it's going to go to the germinal center where it's going to go through somatic hypermutation and selection through antigen-mediated selection. What is required for germinal center formation? You do need CD40 ligand interactions in order to get germinal centers to actually form properly. What happens in the germinal center? So we get proliferation of the B cell and somatic hypermutation, and this is where you're going to get a B cell ultimately with a higher affinity binding antibodies. And you also get class switch occurring in the germinal center after it comes in contact with antigen. Which cytokines promote which isotypes of antibody? So there are some cytokines listed on the left here that promote different isotypes to be made. So IL-4 will help promote IgG1 and IgE. TGF-beta helps promote IgA and IgG to be IL-5 promotes IgA, and interferon gamma promotes IgG3 and IgG2A. What is different between naive and memory B cell responses? So a naive B cell takes about 4 to 7 days to respond. It makes a lot of IgM, and it's short-lived. Whereas a memory B cell only takes 1 to 3 days to respond. It's going to make a lot more antibodies than the first time. So it makes 10 to 1,000 times higher of a peak antibody response. And IgG is predominant, and it might be IgA if it's in the mucosal tissue. So IgM is made first from naive B cells that are activated. Memory B cells have already class switched to either IgG or IgA, and so that's why they wouldn't make predominantly IgM. You also get a lot higher affinity for the antibody, and these are long-lived B cells. Do you get memory develop from all of the B cell subsets? So no, you get the most memory from those T-dependent antigens. So you get memory from T-dependent antigens, but not as much from the T-independent antigens. What B cell subset can generate B cells that make all of the isotypes? And so that are the conventional follicular B2 B cells that we've talked the most about. The B1 cells that exist in the pleural cavities and the marginal zone B cells make mainly IgM. What is important for inhibiting B cells? So to downregulate B cells, negative signaling occurs through CD22, which results in this phosphatase, the SHP1 tyrosine phosphatase. You also can have negative signaling through CD32, which is an Fc gamma receptor, and it has these ITIMs, or immunotyrosine-based inhibitory motifs, and phosphatase are also involved in this process. CD5 is a negative regulator for B cell signaling. Many of the CD5 positive B cells make IL-10, and IL-10 is an anti-inflammatory cytokine, and it can overall be a negative regulator of B cells. What immunoglobulin is present the most in the serum? Which immunoglobulins activate complement, and which immunoglobulins cross the placenta? So the one that's most present in the serum is IgG, and in particular the IgG1. The one that activates complement are the IgGs and IgM, so mainly IgG1, IgG2, and IgG3, as well as IgM. The ones that can cross the placenta are the IgGs, not the others. What are the effects of antibodies, and which antibodies can perform each function? So we have the functions of the antibodies here. So they're all good at neutralizing pathogens and toxins, so basically inhibiting the pathogen from having an effect. They're good at agglutinating, so clumping them all together. The ones that are better at agglutination are IgA and IgM, because the IgA is a dimer and IgM is a pentamer. Some of the antibodies are able to be opsonins and enhance opsonization or enhance phagocytosis of pathogens. IgG is the best as an opsonin. However, IgG can do most of these, whereas some of the others are only good at neutralizing. So depending on the antibody, it can have a better ability to have one of these functions. Complement can be activated by IgG or IgM through the classical pathway. to lyse bacteria. Some antibodies, such as IgG, is important for recognizing something that's wrong on a cell. So an infected or tumor cell can be recognized by antibody, and that immunoglobulin can bind then to an Fc receptor on a natural killer cell, and this natural killer cell can then detect that and kill this cell that's infected by antibody-dependent cell-mediated cytotoxicity. You can also have degranulation of cells activated by antibody binding to a pathogen. So that's something that's very big and hard to get rid of, but this degranulation can release toxic mediators to harm that pathogen. What are Fc receptor mediated effects of antibodies? So if you look at some of the main cells that have Fc receptors, mast cells have Fc epsilon receptors that can bind to IgE and bind to pathogen to cause the degranulation. Opsonization can be enhanced by IgG binding to Fc gamma receptors, so enhance the eating and destroying of pathogens by a phagocyte through antibody bound to the antigen, and then the antibody also binding to the Fc receptor. You can also get the movement of immunoglobulins, and one location is in the placenta. As an example of IgG can go through the placenta through being transported with the help of Fc receptors. You can have the killing of cells that are infected through antibody-dependent cell-mediated cytotoxicity, and how the antibody bound to this Fc receptor then induces this natural killer to kill this cell. And you can also have the movement of immunoglobulins from where they're made in the lamina propria, ultimately into the lumen. So IgA can be secreted by binding to the poly-Ig receptor and being then transported into the lumen, and it can exist as a dimer in the lumen. How are CTLs generated? So CTLs, those cytotoxic T lymphocytes, are made when they get three signals. Signal 1 through its T cell receptor binding antigen and MHC class 1. Signal 2 through the co-stimulation, the CD28 on the T cell binding to B7 molecules. And signal 3 from IL-2, which can come from T helper cells that are activated. And this can happen sequentially or simultaneously. How do they kill? So how do those cytotoxic T lymphocytes actually kill? There's two main mechanisms. They can kill through perforin and granzyme pathways. Perforin basically pokes holes in the surface of a cell when it's released, and granzyme are proteases which can help destroy the target cell. And there's the Fas ligand pathway. So Fas ligand can bind to Fas, which can activate caspases. And caspases are proteins that exist in the cell in inactive forms, and they are active. They can become active and ultimately generate apoptosis or programmed cell death of a given cell. And so that's how cytotoxic T lymphocytes can kill infected cells or tumor cells. What kind of cells do natural killer cells kill? So natural killer cells are another type of cell that can detect a cell that is a host cell, and it can destroy it, but it's one that's pathogen infected or abnormal tumor cell. And so similar to the cytotoxic T cell, it's recognizing a host cell that has something wrong with it. It doesn't recognize it in the same mechanism or same method, but it in the end, the goal is the same, the type of cell that it's looking for. And what determines if a natural killer cell is actually going to kill can be antibody-dependent cell-mediated cytotoxicity. So natural killer cells will kill cells that So if this cell has an antibody bound to it because it has a tumor antigen on it that's recognized here by this antibody can be bound to the Fc receptor, which will then activate this natural killer cell to kill this tumor cell. Natural killer cells can be also activated by this balance of inhibitory and activating signals. And a natural killer cell that's not going to kill a host cell will get more inhibitory signals than activating. And so inhibitory signals come from MHC class 1 binding to inhibitory receptors on natural killer cells. And there can be activating signals come from a host cell that come from stress, and they can be increased in numbers if a cell is stressed. So this cell over here on the left isn't stressed. This is a healthy cell, and so overall this natural killer cell gets an inhibitory signal and it's going to not kill this host cell. On the right here we have two examples of where a natural killer cell will be activated to kill a host cell. So if the host cell is infected, sometimes there's a loss of the MHC molecules on the surface of that cell. And so because the MHC molecules aren't there, they're not able to provide a negative signal to this natural killer cell. And this natural killer cell will only see an activating signal, and it will ultimately kill this host cell by the release of perforin and granzyme. You can also get the killing of a cell even though MHC class 1 is there if there's an increase in the amount of activating signals. They can increase on cells that are under stress, and it can overall provide this natural killer cell an activating signal to ultimately kill this host cell. So what is common at barriers? So if we look at some of the main things, there tend to be one or more layer of epithelial cells. So epithelial cells are common at many different barrier sites. Having a microbiome, so microbes that exist there, is common. And the production of antimicrobial peptides is common. And signaling to immune cells if there's damage to that epithelium is common. Where are these lymphoid tissues, or with respect to the MALT and the BALT and those things? So we have lymphoid tissues that aren't quite a lymph node, but lymphoid tissues in many of the mucosal sites or barriers. And we do have lymph nodes in many of these sites as well. So throughout, we do have many different ways to access adaptive immune cells. So these are locations where you're going to see a lot of these lymphocytes localized. What is in a delicate balance in the gut? And so if you think of the gut, you're taking in all kinds of foreign material because you're eating all the time. You don't want to respond to those things that you eat. You want to be tolerant of them, but you do want to respond when you have pathogen coming into the gut. So in your different barriers, you want to have homeostasis. You want to have the overall response to be tolerant so that you don't have inflammation unnecessarily. If you don't have a good homeostatic balance, you can get inflammation, and you can get inflammation in response to antigens that are pathogens, and you can ultimately activate Th1 and Th17 cells and innate lymphoid cells to then recruit other cells, and including granulocytes, to help try to remove any pathogens that are there. So there's this constant balance between being tolerant and being inflamed in the gut. Where in the gut is there more microbial diversity? If you look in the gut, there is more microbial numbers and diversity in the large intestine compared to the small intestine. What is in the lamina propria? The lamina propria has the majority of the immune cells in it. The lamina propria is below that epithelial cell layer. How does IgA get into the lumen of the gut? So IgA is made in the lamina propria from plasma cells that are secreting IgA. So it's made as a dimer because it has this J chain that attaches it. It interacts at the basolateral side of the epithelium with the poly-Ig receptor, and it then is moved through that epithelial cell and released on the other side with the what's called secretory component. So part of the poly-Ig receptor comes off with that IgA and helps it stay stable. What do M cells and goblet cells do? M cells are important for the movement of antigen across the epithelium, and so are goblet cells. They can also transport antigens into the lamina propria. Which cells in the gut make mucus? So it's made by goblet and Paneth cells. How are antigens detected in the gut? So they're basically detected because when they're delivered into the lamina propria, and they can be delivered through M cells, transporting them from the lumen to the lamina propria. They can also come in through binding to an antibody that's bound to an Fc receptor and be brought in. They can be brought in through goblet cells, and they can also be brought in by antigen-presenting cells that basically reach out through the epithelium and then grab onto antigens that are there and sample what's in the lumen. How do commensals promote tolerance? So the microbes that exist in the normal flora in the gut, they can help promote tolerance because they can activate the production of Reg3. So it's one way, the expression of Reg3, and they can also have short-chain fatty acids that can help activate Treg cells. PSA from Bacteroides fragilis can activate Treg cells as well. So overall, these normal flora can help promote this tolerogenic response. They can help promote the production of IgA, which then, when it exists in the lumen, can help neutralize pathogens that are there so that they can't even get in past that barrier. They are able to help promote the Treg cells that are there by promoting more of an IL-10 response. So IL-10 is an anti-inflammatory cytokine which promotes further Treg cells to be generated. How is IgA made in the gut? So you can get IgA generated from T cell dependent antigens or T cell independent. And when antigens come in, they can be recognized by the B cell through the B cell receptor. T cells can help give another signal, and given the cytokines that are in the area, such as TGF-beta, it'll promote an IgA producing plasma cell. If you don't have a T cell response, so you have T cell independent production of IgA, you get B cells that are activated through the B cell receptor and also have these factors that are made, such as APRIL and BAFF, that help kind of promote the production of a plasma cell that's going to make IgA. What happens when there's disruption in the gut? So if you have more of these pathobionts or microbes that are considered to be not peacekeeping or more invaders, you can get damage to the epithelium and ultimately inflammation. And if you have inflammation long term and a disruption to the microbiome, that seems to be a factor in inflammatory bowel diseases. What is different in the airway compared to the gut? In the airway, you have cilia on your epithelial cells. You also have club cells, which are unique and secretory cells. You have dust cells, which are alveolar macrophages. And then you have lymphoid tissues that are there, which have some similarities to Peyer's patches that exist in the gut. They just structurally look a bit different. Is there or are there commensals in the respiratory tract? There are commensals, it seems, throughout the GI tract. The respiratory tract is different in that in the upper respiratory tract you do have a normal microbiota, but in the lower respiratory tract, so from the trachea down, it seems to be sterile and you don't have a normal microbiota there. How does the immune cells respond to pathogen in the airway? So depending on the pathogen, the response may be different. If you have bacteria, fungi, viruses, they will signal through pattern recognition receptors, and they will promote the production of certain cytokines depending on the pathogen. That will promote activation of an extracellular response or intracellular, and activating those cytotoxic T cells to kill infected cells and ultimately to make cytokines that will help clear the pathogen. Interferon gamma helps make macrophages, for example, work better and get rid of pathogens. IL-17 and IL-22 are helpful to make promote cells to make antimicrobial peptides and other chemokines that will promote the recruitment of other cells to the area that can help clear the pathogen as well. If it's a worm or allergen, you may get damage to the epithelium, and these alarmins produced, such as IL-25 and IL-33 and TSLP, and you'll get more Th2 cells or Th9 and some of these innate lymphoid cells as well. And the types of cytokines that you'll have activated will promote, in the end, the production of mucus. And if it's a worm, the killing of the worm, which will be helpful. Mast cells, when they're activated, they can help recruit other cells to the site as well. What's different about the skin as a barrier, and what are Langerhans cells? If we look at the skin, there are multiple layers of epithelium, so that in and of itself is different than the respiratory tract and the gut. We have a thick layer of keratinocytes. In here, the Langerhans cells are dendritic cells that are given a special name that exist in the epidermis. How do naive T cells get into the lymph node? So they circulate through the blood and they get into the lymph node through high endothelial venules, which are expressing ligands for L-selectin that helps slow them down. L-selectin is expressed on these T cells that are naive, and they will, when they slow down because of this interaction of these cell adhesion molecules, they will be activated by the chemokines that are made in this area. So CCL19 and CCL21 will bind to CCR7 to help these T cells to go into the right spot in the lymph node. They will actually extravasate through. So this whole process is called extravasation. It's initiated by those selectins. So L-selectin helps them get into the lymph node. Chemokines activate them to stop, and then the chemokine binding to their receptor promote integrins to bind with high affinity to really adhere tightly to these endothelium, and then ultimately diapedese through the endothelium. Are there high endothelial venules in the spleen? No, they're not. And the blood seems to kind of deliver cells in here in this marginal sinus. There are chemokines there that help bring them into the area, but overall selectins and L-selectins don't seem to be involved in bringing lymphocytes to the spleen where they do in the lymph node. How do B cells get into the right place in the lymph node? B cells get into the right place because there are follicular dendritic cells that are making CXCL13, so that's a chemokine that binds to CXCR5, which is on B cells, and so that helps bring the B cell to the follicle, to the right spot there. How do lymphocytes get out of the lymph node? So we're revisiting this. We've already looked at how lymphocytes who get into the lymph node, if they've been there for, I think, 18 or 21 hours, something like that, so they've been there a while, haven't found an antigen, they'll start to upregulate their sphingosine 1 phosphate 1 receptor, and they will, when they increase the receptor expression, that means they can bind to the sphingosine 1 phosphate, which helps them then move out of the lymph node. How does antigen get to T and B cells? So antigen can be brought to the lymph node through dendritic cells that migrate there, that have captured antigen, migrated to the lymph node, processed the antigens, and then present them. They can ultimately bring them in through these dendritic cells moving in. They can also be delivered to B cells by moving the antigens to macrophages that are there in the B cell follicle or on the edge, and they can then be passed off to B cells. Exogenous antigen or soluble antigen can also get into the lymph node from blood, and the afferent lymphatics as well. And then they can go, and they kind of tend to go to the B cell follicle location. So macrophages there will take them up and bring them in to the B cell follicle. Where do T helper cells help B cells? So they help them at this border of the follicle, and this is because when B cells get activated, they will increase their CCR7, which then allows them to be kind of attracted to the T cell zone of the lymph node, and so it moves them to the border, and then they can there interact with T cells. What happens in the germinal center? So in the germinal center, this is where you get somatic hypermutation, so you can get ultimately, hopefully, antibodies that bind with a higher affinity to antigen. How are CD8 positive T cells activated? They seem to have this 3-cell interaction between an antigen-presenting cell and a T helper cell and the CD8 positive T cell. It gets signals from the dendritic cell as well as from the T helper cell to ultimately make that CD8 positive cell active. Where do the effector cells go? They can go, if it's a T cell, to the different organs and sites of infection, and it relies on the production of chemokines in those different sites to help guide it to know where to go. Plasma cells can go to the lymph node, the middle part, the medulla, to make IgM, but IgG producers will often move to the bone marrow, and IgA will go to the malt and then make their antibodies there. How is the immune response regulated? So it seems to contract with about 2 weeks after activation, and reducing the survival signals like IL-2, IL-7, and IL-15 can help regulate them. They also can be induced to die by apoptosis through fast ligand signaling, and also inhibitory signals from CTLA4 or PD1, as well as inhibitory cytokines like IL-10. Where do memory cells go? So depending on the memory cell location, we tend to give it a different name. So it's a central memory cell if it stays in the secondary lymphoid tissues. It's an effector memory cell if it circulates throughout tissues in the periphery. And if it's a resident of a tissue, we tend to call it a TRM, so a long-term resident of a given area. How do activated lymphocytes stop circulating around lymphatics? So when a T cell is being recruited, just moving through and looking for antigen, it's going to be recruited into the lymph node because it has L-selectin on its surface and CCR7, and it will be recruited to the lymph node to look for antigen. If it doesn't find one, it will, through interactions with S1P1, then leave through the efferent lymphatics. If it does bind to antigen and it proliferates, or if it's a memory cell that's coming in and being activated, it will, these cells, as they leave, they decrease their expression of L-selectin, and they increase other selectins and other chemokine receptors, which will help them move to sites of inflammation. So the types of selectins and chemokine receptors on the surface of the effector cells will be different so that they will move into the tissues that they need to be in. So I hope that this video was helpful for you as a review. Please note that anything that was mentioned in class may be tested.